Hey guys, it's me, Mr. Plazik. This video series is going to deal with your biology text, and in particular Unit 7.3, which is dealing with cell transport. So in this, what we're going to talk about is how a cell is able to get substances to come into it and how a cell is to be able to get substances to leave it, both using energy and not using energy at all. So let's take a look at the cell membrane itself, okay? So the point of a cell membrane is to be able to separate the inside of the cell from the outside of the cell. So remember, like with homeostasis that we talked about, is we must maintain internal balance. Well, a cell must maintain internal balance as well. It must be able to separate the internal chemical environment of it from the outside chemical environment that it's currently present in. So it has to maintain a certain glucose concentration, a certain sodium concentration. It might have to contain a certain pH that's different from that external environment. And the cell membrane is what's going to allow the cell to be able to do that. It separates the contents. It separates what's going on the inside of the cell from what's happening on externally in the external environment. So we have to keep these conditions relatively constant. And how it does this is it controls this by regulating molecules coming in and out of the cell. So it selects what molecules it wants to come in and what molecules it wants to leave the cell in order to maintain homeostasis. When we take a look at the cell membrane of plant and animal cells, it's known as a lipid bilayer. And a lipid bilayer is composed of these little tiny molecules, if you look down here, that we call phospholipids. All right, so these little molecules that are here are called phospholipids. A phospholipid looks something like this. Right here is what a phospholipid looks like. This is a real generic diagram of a phospholipid that I'm drawing right here. But this is a phospholipid, okay, and it's a bilayer, so that means there's going to be two. Okay, there's going to be two of these. And these phospholipids orient themselves in this particular direction itself. So when you take a look at your cell membrane, it's loaded with millions and millions of these phospholipids all around in a circular-like fashion for that particular cell that we're looking at. The phospholipids themselves are composed of a lipid, a fat molecule that it's composed of, and then it has a polar head and a non-polar tail that I'm going to talk about next. So when we take a look at these phospholipids, part of this phospholipid likes water, okay, and they're located on the outside. So when I take a look at a phospholipid that's here, all right, this head region that I'm drawing right now, this is what we call the hydrophilic region. It loves water. And then you have these tail-like structures, what we call hydrophobic. And they hate water. They dislike water, okay, and they're located on the inside of the bilayer. So we can see here, I'll draw another phospholipid here. Remember, this is how they orient themselves. So you have a water-loving region that's right here, all right, and then this middle section that's right here is the hydrophobic section that dislikes water. And this allows the cell to be able to automatically then control what's coming in and out of it to a degree because we have this separation between water-loving molecules and water-hating molecules. So now things can't just easily come right across this particular membrane. Unless if you're super, super small, you have the ability to kind of squeak through this membrane. So remember, take a look, the part that doesn't like water is called hydrophobic. The parts that like water is called hydrophilic. So we have our hydrophilic and we have our hydrophobic regions that we just got done talking about. This prevents water-soluble molecules from passing through the membrane. So again, if I'm a water-soluble molecule, I'm not going to be able to make it through this middle portion, okay, because this middle portion hates water. We kind of think of oil and water don't mix, so we have that repelling that's going to occur from that middle section to prevent things from just making their way freely across the membrane. One exception to this rule, okay, are very small molecules. So if I draw this, again, if I draw this little phospholipid molecule, it's here, and we draw a little phospholipid bilayer, when I take a look at it just like this, all right, we'll draw another one. This is a really bad tail on this thing. Draw another one right here. If I'm a really small molecule, I can squeak through, okay, this bilayer, okay? So if I'm an oxygen molecule that's out here, I can make my way through the cracks here and make my way through into the cell. All right, so one exception is to who can defeat the bilayer is something that's super, super small that can fit through these particular cracks themselves. And these molecules do not have any electrical charges either. So we take a look at a membrane, okay? You have membranes that cover a variety of things within your body. A cell has a membrane that's going to, like I said, control its internal environments from the external environments. Some membranes that we have are permeable membranes. That means that anything can get through this particular membrane. Some membranes are impermeable membranes, all right? When we identify a cell membrane, it's not permeable or impermeable, but it's a mixture between the two. Okay, it's going to allow some things to get in, but not everything to get in. So it's what we call selectively permeable or semi-permeable. So our cell membranes, plant cell membranes, are all what we call selectively permeable membranes. All right, they allow some molecules to pass through where other molecules don't have the ability to get through at all. So that's what we mean when we take a look at cell membranes. They're what we term selectively permeable. So that phospholipid bilayer that we just took a look at is a selectively permeable membrane. Some things can get through, but not everything can get through in a selectively permeable membrane. So the next thing we have to take a look at, now that we've identified, our cell membranes are what we call selectively permeable. Some things can get in, some things can't get in. It's composed of these phospholipids that have a hydrophilic region and a hydrophobic region. We have two types of transport. Remember, this whole unit's dealing with cell transport. How things get into cells, how things get out of cells. And there's two types of transports that we're going to talk about. The first one's called passive transport, and then the second one is called active transport. In passive transport, no ATP is required. No cellular energy is required to be able to move these particular substances across the membrane. It happens on its own, okay? So no ATP, no cellular energy. An active transport is going to require cellular energy in order to get the substances to move across the cell membrane. So we term passive transport is going to move down the concentration gradient. And what I mean by going down the concentration gradient is we're going from a high concentration to a low concentration. I'm going to draw a little picture right here, okay? And let's say that this is a cell right here. And let's say that this cell is permeable to this little glucose molecule that's right here. I'm representing this glucose molecules by these little tiny dots that are right here. And let's say that for all intents and purposes that this cell membrane that I have is permeable to this particular glucose molecule that's here. Now if I take a look here inside the cell, okay, there's not as many glucose molecules as there are outside the cell. So moving down the concentration gradient is going from an area of high to low concentration. So in this particular case, this glucose molecule is going to move into the cell membrane, is where it's going to move. All right, this happens on its own. It goes from an area of high concentration, which I'll represent with an H, to a low concentration, which I'll represent with an L. So moving from high to low concentration, that's what we call going down the concentration gradient. And that's what passive transport is. Passive transport, we're always moving from an area of high concentration to an area of low concentration. The first type of active transport that we're going to deal with is the movement of a substance other than water, okay, across the cell membrane from an area of high concentration to low concentration. All right, we call this diffusion. All right, the molecules really never stop moving, but they go from this high to this low till they're kind of equilibrated, okay, throughout the particular inside of the cell and outside the cell. So the outside of the cell has the same amount of molecules as inside the cell, and that's when we reach this equilibrium. So we go from high to low until the outside is the same as the inside of the cell, and this is what we call diffusion. Now diffusion is dealing with things other than water. So we're dealing with things like oxygen, glucose, sodium would be diffusion, but again, we're moving from high concentration to low concentration as what we're dealing with in diffusion. Now, I'm going to get a little nerdy here with you, okay? We have diffusion, and technically diffusion is just movement across the membrane, okay? So it's something that's, for the most part, is really, really tiny. So I know I said something like glucose a few seconds ago. I said something like sodium and stuff like that. Oxygen is one that diffusion really happens with. Diffusion, oxygen just kind of comes right across that particular membrane. Other molecules have to do something. They have to go through facilitated diffusion. It's still diffusion, Okay, don't get mad at me. Oh, Mr. Plazik, you know, what is facilitated diffusion? Well, facilitated diffusion, we're still moving from a high to low concentration. Okay, we're still moving from a high to low concentration. But the problem being is that, this is my little cell membrane that's right here. The problem being is these molecules might be a little too big, okay, or they might be a little more water soluble, that they can't just freely come across this membrane, so they have to go through what's called a channel. So a facilitated diffusion is when a particular product has to travel through a channel. Right here is a generic channel that's here. So this might be like a glucose molecule that's right here, and this glucose is going to go from high to low concentration. But rather than have to travel through a phospholipid or sneak through this phospholipid bilayer right here, okay, these two little phospholipids, it makes its way through the bilayer by going through a channel. Okay, so facilitated diffusion is going through a channel. All right, you have specific channels inside of your cells that deal with glucose. Okay, you have specific channels that deal with water. When we take a look at the net movement of water, a lot of times it's facilitated diffusion because it's going through a channel, but we call the net movement of water, we don't call it diffusion, we call it osmosis. Okay, we call it osmosis. So if water is moving through a channel from high to low concentration, we call the net movement of water, we call that osmosis. And we're really keyed in on osmosis in this particular unit because the net movement of water is going to cause a cell to expand, or the net movement of waters might cause a cell to shrink, is what we're kind of focusing in on this particular unit. We've done a variety of labs and activities. I did an egg lab with you. We just got done doing a dialysis tubing lab that we took a look at, in which we put, you know, a starch solution in dialysis tubing, and then we expose it to an aqueous environment and trying to figure out which way is water moving. If water's moving into the cell, it's going to cause the cell to expand. If water's moving out of the cell, it's going to cause it to shrink. So know that the net movement of water that we're talking about is what we call osmosis. Facilitated diffusion, okay, is when we have a product that's traveling through a channel. It can't fit directly through a cell, can't fit right directly through the phospholipids themselves. So here we go, osmosis. Osmosis is the net movement of water. Osmosis is the net movement of water, again, from high to low concentration. When you're dealing with problems in this unit, you're always asking yourself, okay, where's there a higher water concentration at? Okay, the water is always going to move from high to low. Here's an example here, okay, of some osmosis problems that we have. We have a semi-permeable membrane. We have the water molecules that are represented by black that are here. And we can see that there's a high amount of water molecules on this side of the membrane. So we're going to go from high to low, okay? And when we go from high to low, okay, it goes through this membrane. This level right here is going to rise, okay? So this level of the chamber here is going to go up, all right? And this level of the chamber is going to go down because it's losing water. And you can see that represented in this particular picture here. Now these little tiny white molecules that are in here are sugar molecules, and in this particular case, this semi-permeable membrane is not permeable to sugar. So notice sugar would normally go from high to low, but because it's impermeable to sugar, sugar is not making its way across the membrane in this direction here because it's an impermeable membrane to sugar. It's permeable to water. Remember, the net movement of water is what we call osmosis. So sometimes, okay, we take a look at the net movement of water in or out of a cell, and this can exert a pressure on the cell that we call osmotic pressure. All right, so sometimes this puts a little bit of pressure on the cell if it expands so much, and we call this osmotic pressure. So let's take a look at some things, I've created this little chart here for you. So if we have a greater concentration of water outside the cell, okay, so let's say that I'll draw a little beaker here. We'll draw a little beaker here. This is my little generic beaker that's right here, all right, and this beaker is loaded with a lot of just water, okay, and I put a cell in here, okay, and this cell, okay, we'll put little tiny sugar molecules in here with the water. All right, so I can see in this particular picture itself, there's a greater water concentration outside the cell than inside the cell. So what's going to happen in this case is water is going to move into the cell. All right, So water would move into the cell in this particular case, and that's what I'm talking about here. And the internal pressure inside that cell is going to increase. Why is that internal pressure going to increase? Well, because you have more water moving in, pressing up against that cell membrane. The opposite of this, okay, the opposite of this would be, okay, let's change things around. Let's say I have a beaker, and this beaker is loaded up with a sugar water. Okay, this beaker is loaded up with a sugar water. Put little bits of sugar in here. They're like this. And I were to put a cell in here. This is my little cell that's right here. And this cell doesn't have any sugar in it. It just has water in it. Okay, water is going to leave the cell. And when it leaves the cell, it's going to cause less pressure to be exerted on the walls. All right, this cell is going to shrink. This would be like a dehydrated cell. But note, water is moving from an area of high concentration to low concentration. Now is when we're going to start making terms, okay, for each of these solutions that we have. We call a solution, okay, a solution that has more water in it, okay, than inside the cell, let's say. Okay, so let's say we have a solution that contains 100% water, and then inside the cell we contain 50% water and 50% glucose. The solution that we're putting the cell in is termed hypotonic. So a hypotonic solution contains a greater quantity of water per se, meaning that it contains more water than the other thing that we're looking at. Hypo, when I say hypo, I mean more H2O. So if I put a cell in a hypotonic solution, a lot of times it's going to expand. Sometimes it'll expand so much, if it's an animal cell, it will bust open and it will lyse, is what will happen. And that's a bad thing. If a cell busts open and bursts, your cell dies inside of you. This could be a situation where you overhydrate yourself. Okay, you drink too much water, and you continue to drink water, and you continue to drink water, and your body can't expel it fast enough. The cells inside your body may bust. Now, in a plant cell, when you put it in a hypotonic solution, it expands, okay? It expands. Remember, a plant cell, though, has a cell wall. Okay, so what happens with a plant cell is you put a plant cell in a pure water solution. That plant cell will expand, okay, but it will press up against that cell wall and allows it to stay nice and firm, is what's going to happen in a plant cell. Another, the opposite end of the spectrum that's here, so we have a hypotonic solution that's right here, hypo more H2O. The opposite of hypotonic is hypertonic. In a hypertonic solution, it contains less water. Okay, so this would be that previous example when I put that cell into a thing of sugar water or a thing of salt water. And what's going to happen? Well, water is going to move from high to low concentration. In this case, in a hypertonic solution, there's less water and there's more solute. Okay, we call this stuff that dissolves in water, we call it solute. This could be like sugar, this could be like salt, other types of molecules that are dissolved in this particular water. Well, if I take one of your cells, okay, that has a higher water environment inside of it, and I put it in a beaker, okay, I put this in a beaker that contains less water, this cell, water is going to leave this particular cell itself, and it's going to cause the cell to shrink. The cell will become dehydrated. The same thing happens with a plant. Now, if they're both equal, okay, let's say that the outside of the cell is the same as the inside of the cell, we're putting in what's called an isotonic solution. In an isotonic solution, yes, you have water movement, but water's moving equally inside the cell, that's moving out of the cell as well. This is in an isotonic solution. So these are the three types of solutions that we have. We have a hypotonic solution, we have an isotonic solution, and we have a hypertonic solution. Hypo, more H2O. Hypertonic solution, less water. Water's going to move out. It's going to make it dehydrated. Again, you can see here we took a red blood cell. We put a red blood cell in three different types of solutions here. I put a red blood cell in an isotonic solution. It stayed the exact same shape. I put a red blood cell in a hypertonic solution. It shrunk in size. I put a red blood cell in a hypotonic solution. Water came rushing in and it expanded, is what's going to happen. Know these three different words. Know hypo, more H2O, isotonic, and hypertonic. Remember, isotonic water concentrations are equal on both sides. You have a net movement of water that's going to be equal. You have an equal amount of water coming in that's leaving the cell. In a hypotonic solution, you have a greater amount of water outside the cell than inside the cell. Therefore, you have water that's going to rush into the cell. This can eventually cause the cell to bust or lyse open. In a hypertonic solution, you're putting a cell in a solution that has less water in it, that cell is going to shrink. All right? This is what happens in each one of these solutions between the two cells. Okay? A plant likes to be in a highly aqueous environment because, remember, it has a cell wall. So when it has that water that can press up against that high water, that cell membrane that can press up against that cell wall, okay, it allows it to stay nice and firm. Okay, you've seen a plant that doesn't have a lot of water in it, okay? Yes, it's still, but it becomes limp, it becomes flaccid, is what's going to happen with that particular plant. So the less water a plant has in it, the less strength that it has because it doesn't have that cell membrane pressing up against the cell wall. Again, exact same picture, trying to explain the exact same thing here with hypotonic solutions that we have there. Now, a plant cell has a vacuole that's associated with it, okay? And this vacuole can take on water as well to help press up against the cell wall, to help the cell maintain its actual firmness as well. So this is kind of a water-filled compartment inside of the cell membrane that's also going to load up with water. That's also going to contribute to the firmness of the plant itself. Now we're going to talk about, so we just got done talking about passive transport. Now we're going to talk about active transport. The key with active transport is active transport requires ATP. It requires cellular energy. So active transport is going to require cellular energy. With active transport, we're not going to go with the concentration gradient. We're going to go against the concentration gradient. We also might have to use active transport if a molecule is super, super big and we don't have a channel that it can fit through, that we have to let things get into the cell or out of the cell. We're still going to have energy to be able to do that as well. So active transport, we're going to go against the concentration gradient is what we're going to do with active transport. An example of this, okay? Let me draw a little example here for you so you kind of understand this. This is a nice little plant here. This is a nice little flower that I'm trying to draw here. Okay, this is my nice little flower here. And this little flower has a root system that's composed of cells. So right here is the root system of the plant. Okay? Now each of these little roots, okay, is a cell, okay? These little roots are composed of hundreds and thousands of cells that are here. We'll draw the soil right here. Let's say that inside the soil here, okay, there's some molecule that's in low quantities that this plant needs. Let's say it's like phosphorus or something like that. But if I take a look inside the plant cell, okay, there's a ton of this phosphorus that's inside this little plant cell that's right here. There's loaded up with phosphorus. But this plant needs this phosphorus molecule, all right, in order for it to maintain its metabolic activities. Granted, okay, if this were going to go through passive transport, it'd go from high to low, but this plant really wants this phosphorus that's out here, so it's going to take it from a low concentration and move it into the plant into a high concentration. That would be an example of active transport, okay? Although it's in low quantities in the external environment, your body still needs it, or the plant still needs it to be able to take in, so it's going to move it across that concentration gradient and go from low to high. But this just doesn't happen naturally, okay? This just doesn't happen naturally. It just doesn't naturally, so you have to put energy into this system. So that's what active transport is. A lot of times we're going against the concentration gradient in active transport. Another thing that we have inside of our bodies is we have pumps. We have sodium-potassium pumps is one thing that we have inside of our bodies, okay, that utilize this active transport. If you guys take me for human anatomy phys, I talk about a sodium-potassium pump that's used to generate action potentials, to generate electrical impulses in neurons, in which we're moving sodium and potassium across the concentration gradients against the concentration gradient is what we're doing in active transport. Right here is that sodium-potassium pump that I'm talking about. So you can see in this particular situation, we're kicking 3 sodium out and bringing 2 potassium in. Even though that there might be a higher quantity of potassium inside the cell, we still want to bring 2 potassium in and kick 3 sodium out, even though that sodium's in a higher concentration outside of the cell. That's an example of active transport. So in active transport, you kind of have to take the realization that, hey, there's something that's in the external environment that my body wants to take away from it, even though it's in a low concentration. We want to move it from a low concentration to a high concentration. That's what active transport is. Another thing that we have is bulk transport. So sometimes a molecule is so super large that it can't just fit through a channel. It can't just fit through one of those little cracks in the phospholipids. So therefore we have to go through bulk transport. And this requires energy as well, because we're going to build something called a vesicle to transport this substance inside the cell or outside the cell. So the two types of bulk transport that we have, one is endocytosis, which is bringing material into the cell. And the other one that we have is exocytosis, which is making a product and then exporting it outside of the cell, is what exocytosis is going to be. We normally just package these things in little tiny bubbles we call vesicles, is what we're going to do. Here's the two types of endocytosis. We have phagocytosis, which is like cell eating, when a cell comes and it just kind of surrounds something and it engulfs it basically with a vacuole. And then pinocytosis is like cell drinking. So those are two types of endocytosis that we have. In exocytosis, okay, so this would be like, this right here is your cell eating. This right here, you can see the cell membrane surrounding this product and bring it in a vesicle. This would be your pinocytosis right here, is cell drinking, which is cell surrounding some type of liquid medium and then bringing this in. Again, we're bringing it into the cell in a vesicle. This is another form of active transport. Even though we still may be going from high to low concentration, we have to create a vesicle, we have to surround it, and we have to bring it in to the actual cell itself. Exocytosis is when we're going to create a vesicle inside the cell and then release it to the external environment, is what we're doing with exocytosis. So think endocytosis coming in, exocytosis leaving. So that was about 20-some minutes, guys, of cell transport. Hope you enjoyed the video. Hope you learned something from it. If you have any questions, don't hesitate to come see me in class prior to taking the test. This is a cell, okay, this is an egg that we put in a corn syrup solution. And you can see, if you look real close at this egg itself, this egg shrunk. Okay, so this egg, if we can get a good glimpse of this egg, that's right there, this egg shrunk. And this egg here was put in a solution that was what we call hypotonic. And you can see that it expanded, so a whole bunch of water rushed into the cell at that particular point. Like I said, guys, good luck with studying, and don't hesitate to see me.